Good morning and good afternoon. This is Jonathan Willmer presenting on behalf of the ATA188 study investigators the results of a phase 1 study of ATA188, an off-the-shelf allogeneic Epstein-Barr virus targeted T-cell immunotherapy for progressive forms of multiple sclerosis. Many studies have shown that Epstein-Barr virus infection, particularly in B cells, is strongly involved with the pathogenesis of MS. ATA188 is an investigational off-the-shelf allogeneic T-cell immunotherapy targeting EBV-infected cells, and the inventory is sourced and produced from unrelated EBV seropositive immunologically diverse donors. This slide shows the detail of the manufacturing process and can be reviewed in the full poster. EBV-targeted T cells are produced and then characterized by EBV-targeted reactivity, HLA restriction, and then cryopreserved. Potential patients are HLA typed, and ATA cell product is selected based on HLA and allele profile. The methods of this study have been previously reported. Part 1 of the study evaluated safety and potential efficacy and involved 4 dose escalation cohorts where each subject was treated with 2 cycles of ATA188 and followed for 12 months. Subjects completing 12 months were eligible for a 4-year open label extension with annual treatment. Endpoints are shown in this slide, and in this presentation we present the results of efficacy showing sustained disability improvement as defined by the table in this slide. Here we report data on 24 subjects from the 12-month dose escalation portion of the trial, 16 of whom entered the open label extension and have 15 or more months of data available as of October 2020. The results are shown here, and baseline characteristics have been previously reported. Safety has been previously reported, but the safety profile of all 4 cohorts has been deemed to be acceptable. The results of disability improvement are shown in the 2 graphs in the middle of this slide. Sustained disability improvement is shown on the left. There is a dose-related increase in subjects per cohort exhibiting SDI over 12 months, and this is shown to be driven largely by EDSS improvement over 15 months, as shown on the right side of the right graph on this slide. In addition, SDI responders from cohorts 1 to 4 with SDI at 12 months versus those without tended to have greater improvements in fatigue severity scale, MS walking scale, and MS impact scale scores, as shown in the boxed charts on this slide. This table shows the results of all patients achieving SDI. The color codes explain the trends for disability improvement, green being improvement and pink being decline. The yellow boxes indicate when the patients were redosed, and although patients were supposed to have been redosed at 12 months, a few patients were dosed late due to logistical issues at the sites and COVID. And one subject, the first one in row A, did not enter the open label extension but achieved SDI at 6 and 12 months. For completeness, this table shows all patients who entered open label extension who did not achieve SDI, and this data can be reviewed in the full poster. In summary, in the results, data from the part 1 of the trial demonstrated the safety of ATA188 at the doses studied and provided signals to support proceeding to the randomized control portion of the trial. Beneficial effects were observed on clinical parameters, including disability, with higher doses associated with higher rates of sustained disability improvement. There was a dose-related increase in the number of subjects meeting SDI criteria. In the 2 highest dose cohorts, 42% and 50% met SDI criteria at 12 and 15 months respectively. And at 12 months, subjects who met SDI also tended to show improvements in fatigue, physical function, and MS walking scale at 12 months compared to those without. And specifically for the open label extension, 16 subjects entered and had data beyond 12 months. 6 of the 7 subjects who met SDI criteria at 12 months entered the open label extension and all maintained SDI at future time points. Although encouraging, a randomized placebo-controlled trial is currently in progress to confirm these results from the open label portion of the trial. Initially, the study was started at the cohort 3 dose, but these data have enabled us to move to the cohort 4 dose with the next protocol amendment. This slide shows our disclosures, and thank you for listening.